back to humans, to healthy humans, and looking at the relationship between hippocampal subfield volumes and navigation ability. And that's work that's been completed by Alina too and Elizabeth Chrastil. And so I am not sure which of the two are presenting, Alina or Elizabeth, but the floor is yours.
Thank you so much. Yeah, it's going to be me today. So I'll just go ahead and share my screen. All right, so thank you so much for that amazing introduction. Again, my name is Alina too, and I work with Dr. Elizabeth Chrastil in the Spatial Neuroscience Lab at the University of California, Irvine. And before I begin, I just want to give a big thank you to the Spatial Cognition for hosting this wonderful virtual event. And all the talks before mine have been super insightful and amazing. And I'm really honored to be here to speak about my research, which looks at the relationship between hippocampal subfield volumes and individual differences in human spatial navigation. To start off with some background, navigation, I think we can all agree, is really critical for survival. As we see in animals, like what we just heard about rainforest frogs, animals need to be able to navigate their environments for food and water and reproduction, but they also have to navigate back home safely without getting lost or potentially killed. So for animals, navigation is really critical for survival, but what about humans? For humans, modern humans have to still be able to navigate their environment to survive. So we're not getting picked up or dropped off at a different location, but we do drive around, we hike, we travel, etc. And we have to also be able to find food, shelter, etc. to be able to survive. However, we see a large variation in navigation ability in humans. So some individuals are really great navigators and they can learn new environments easily, but others like myself struggle to navigate really familiar environments even when I rely on a GPS. So a big question is, why is there such big individual differences in human navigation? My central research question is, do volumes of brain regions correlate with the ability to successfully learn and move in a novel environment? In particular, we're interested in looking at the hippocampus and other medial temporal lobe regions, as these regions have been shown to relate in some aspects to learning, memory, or spatial ability. And of course, all of these are really important for successful navigation. So far, what we know about the relationship between navigation and the hippocampus is McGuire and colleagues in the early 2000s studied London taxi cab drivers because they were known to be expert navigators with decades of experience navigating through London's really complex and intricate routes. So when compared to control participants, these taxi cab drivers are found to have larger right posterior hippocampi in terms of volume. On the other hand, when we're looking at the other population, including patients with mild cognitive impairment, patients with Alzheimer's, individuals who have brain lesions, or even older adults who experience aging effects in the brain, these individuals not only have poor spatial ability, but they also have a reduction in their total hippocampal volume. So as you can see in this figure on the right, the left brain is a brain of an individual with Alzheimer's disease, and the right brain is a brain of an individual without Alzheimer's disease. And there's a stark difference between the two, where the left brain has shrunk significantly. So given these findings and a lot more that have shown some relationship between hippocampal volume and navigation, this proposes a hypothesis that shows a correlation between these two variables. And in smaller studies, specifically in the young adult population, there are some relationships that have been seen between hippocampal volume and navigation ability. However, in larger samples, there's kind of been a disconnect where these larger sample size studies show no correlations between total hippocampal volume and navigation ability when specifically looking at the young adult healthy population. So this provides a big motivation for my study, as we're also looking at the young adult population, but we're also going to dive in deeper. So a lot of previous literature has looked at the hippocampus and its volume as a whole, but we want to look at subfields within the hippocampus. So some subfields of interest within the medial temporal lobe include the hippocampus subfields, and these 4 subfields that we're looking at include the CA1, the combined CA2-3 region, the dentate gyrus, and the subiculum. And for other medial temporal lobe regions that we're interested, it includes the entorhinal cortex, the perirhinal cortex, and parahippocampal cortex. So visually and structurally, what that looks like in the brain is shown in this key. So this key, we have on the left column, the 4 hippocampal subfields that we're interested in, as well as the 3 relevant and nearby medial temporal lobe regions. Below the key, we have a 3D rendering of the segmented hippocampus and the segmented medial temporal lobe just generally. Below that, we have a chronoslice. So a chronoslice is as if you were looking at a 2D plane in front of you, but then like move that plane forward and back. That would be a chronoslice of the brain, where the right and left sides would be where your ears are over here. And then a zoomed in view of the hippocampus is shown down below, where we see a segmented hippocampus based on these subfield regions. And since we're able to move the slice forward and back, we can see the anterior, or the more front part of the medial temporal lobe, and the posterior, which is the more like back part of the medial temporal lobe. And one other way to orient ourselves is looking at the sagittal slice, where we see this is going to be the front of the brain, this is the top, here's the spine and the cerebellum. And again, this is just the structure in a different view. So now that we have a general understanding of what these subfields look like in the brain, I'll go ahead and explain how these subfields relate to learning, memory, or spatial ability. So far, what we know is that the CA1 and subiculum volumes have been shown to relate to delayed recall memory ability. And this is, of course, essential for navigation. As an individual navigates a new environment, they have to be able to recall the paths or landmarks that they find along the way. Also, the right CA2-3 volumes have been shown to generally correlate with navigation in young adults, and that's going to be important for our work as we're also looking at the young adult population. The CA2-3 and dentate gyrus volumes not only relate to delayed recall memory ability, but also relate to pattern separation. So pattern separation is the ability to compartmentalize similar sensory inputs and then encode them into distinct, separate experiences in our memory. And this is especially important for our work since we have our participants go through a maze task. And as you can imagine, a maze has very similar looking routes and corners and turns. So it'd be really important to be able to separate out these experiences to be successful at navigating in the maze. So these are the hippocampal subfield regions of interest, but other relevant medial temporal lobe regions include the entorhinal cortex, which has consistently been shown to have grid cells and place cells. The perirhinal cortex activity relates to memory and visual perception. And lastly, the parahippocampal cortex activity has been shown to significantly correlate with scene processing. So given that all of these subfields have been shown to relate in some way to these processes, we do predict that these subfield volumes will be important for navigation ability. Specifically for our hypothesis, assuming that pattern separation is important, especially navigating in a maze task, we predict that the CA3 and the dentate gyrus subfield volumes will correlate with navigation performance. If not, we still predict that the CA1, the CA2-3, and the dentate gyrus subfield volumes will correlate with navigation ability because of their relationship with memory. And otherwise, we will assume the null, which is that there is no relationship seen between subfield volume and navigation in the young adult population. So to test our hypothesis, we had 107 participants complete the maze task. And out of the 107, we had 28 participants where we collected their high-resolution T2 MRI scan, which will be important for our analyses that I will be describing a little bit later. So out of these 28, we had 3 participants who were excluded due to movement in the scanner, which resulted in poor quality of data. So we ended up with 25 participants who completed the maze task, and we have their high-resolution T2 MRI scans. And for these 25 participants, their mean age was around 21 years old. As for the task, this was a desktop virtual maze environment that the participant navigated in. And here we see an overview of the maze, and the red circles within the maze represent objects. So participants were told to not only find the locations of these objects, but also remember where they're located. Also, we have 4 purple lines along the maze walls, and these indicate paintings that were hung on the maze walls that participants could use as a reference point. This is what the participants saw when they were navigating through this desktop virtual maze. And they were able to see the arrow keys that represent which directions they can move in. So they had 3 options: forward, turn right, turn left. But in this case, because of their location in the maze, they could either move forward or turn right. And in the center, we see 1 out of the 4 paintings hung on the maze walls. So participants were given time to explore the maze, but after exploration phase, they were tested on their knowledge of the target objects. During the test phase, for every trial, participants were shown 3 screens. The first screen is a start at object, where the participants were told to start at 1 out of the 9 target objects. The next screen is a go to object, where they were told to go to another. And lastly was the navigation time, where they were able to move around again with the arrow keys. However, all of the objects within the maze turned into red spheres to prevent feedback. So participants didn't actually know if they reached the end goal. They would just navigate to what they think would be going from the guitar to the chicken in this example. So once that was done, we were able to obtain our measure of navigation, which looks at accuracy. And accuracy is calculated by taking the proportion of correct trials and dividing that over the number of total trials. Once they were done with the maze, they filled out some questionnaires that obtained some data about their previous experience with video games and other objective and self-reported measures of navigation as well. So this concludes the measures for navigation that we obtained from the participants, but we also wanted to collect some brain data. This is why we had the participants go through the magnetic resonance imaging, or MRI scanner. And we collected many scans, but some that are of interest for this analysis include the whole brain T1 weighted and T2 weighted scans. So this is for all participants. We have the T1 and the T2 weighted scans. Down below, we see a figure that shows what a T1 and a T2 image looks like. Nextly, for the 28 participants, we had collected their high resolution T2 scans, and this was taken only looking at the mediotemporal lobe, or only focusing on the mediotemporal lobe regions, and the voxel sizes were a bit smaller in the coronal view, so we were able to see at a higher resolution. As for image processing, we use a machine learning pipeline called the automatic segmentation of hippocampal subfields, otherwise known as ASHES. And ASHES takes these brain scans, specifically the high-resolution T2 MRI scan, as an input to then automatically output this image down below, where we see each color representing a specific subfield region. Once we were able to obtain the output that was automatically segmented, we go back through every single slice of every participant's brain to manually edit these segmentations to make sure that the borders are precise and that they're all consistent within participant and also across all participants. So this was done on a software called ITK Snap, where we were also able to obtain the subfield volumes. So this was one of our measures for cortical size, which is volumes, and that's just adding up the total number of voxels that a subfield takes up in the MRI space. And then we also obtain thickness, which is dividing the volume by the number of slices that we see these subfields on. So now that we have our measurements for both navigation and cortical size, this can be used for our analyses. So firstly, just general analyses, we wanted to look at the correlations between these navigation measures. So again, we have both the self-reported and objective measures from looking at the maze accuracy, but also the rest of the questionnaire data. And then for our main question, which is trying to see if there's a relationship between hippocampal subfield volumes and navigation ability, we did partial Pearson correlations between these two variables. And for all of these analyses, we added sex, age, and total intracranial volume as covariates. And then during our data analyses, we noticed that 12 of the 25 participants had premature cutoffs in their high resolution T2 scans. So because of that, we have 13 participants for our correlations between navigation ability and total and subfield volumes within the medial temporal lobe. And then we also wanted to look at thickness. So we had the 25 participants where we correlated navigation accuracy with hippocampal subfield thicknesses. And lastly, we also looked at the other MTL subfields. So that's the entorhinal, the perirhinal, and parahippocampal cortices and their thicknesses. For all of these, we tested these against a Bonferroni corrected significance level of 0.0036. So this is taking into account the 7 brain regions split by hemisphere. And then we did some exploratory analyses of sex differences. So to start off with our main analyses and our main results with the partial correlations between navigation measures and our cortical size measures. Firstly, let's look at the total hippocampal or total medial temporal lobe volumes. So this is essentially our null hypothesis, and taking into account sex, age, and total intracranial volume, what we see here is a plot where we have the total hippocampal volume on the x-axis, and the proportion corrects from the maze accuracy score on the y. And there is no correlation between these two variables, and it has a very large spread. So just to clarify, the total hippocampal volume includes the CA1, CA23, DG, and subiculum. But adding the 3 other regions of interest to make up the total MTL volume, we have that on the x-axis with proportion correct on the y. And again, we see no correlation between these two variables with a really large spread. So these findings are consistent with the studies with larger sample sizes, but we want to dive deeper and look at the volumes of hippocampal subfields individually. So here I have the key again, so you can see the subfield regions of interest as well as the chrono slices with the segmentations on it. And now we're going to look at maze accuracy and its relationship with subfield volumes. So again, taking into account sex, age, and total intracranial volume, this is the table that we have with our results, where the left column is a list of the 7 subfields of interest, and then this is split by hemisphere, so the left and the right hemisphere. We also have the correlation coefficients for these correlations as well as their respective p-values. So here again, our sample size is 13, but the regions that were the regions where their volumes were significantly correlated with maze accuracy were these ones. So the left CA1, the left and right dentate gyrus, and the left and right parahippocampal cortex. So if you remember from my hypothesis, we did predict that this CA1 and the DG regions would be significantly related with navigation ability, but we didn't predict the left and right parahippocampal cortex volume to be significantly related to maze accuracy. And while it's important in scene processing, which is probably crucial for navigation success in our maze task, there's not a lot of literature that looked at parahippocampal cortex volumes and their relationship with maze accuracy. So this can provide more insight and more research in studying parahippocampal cortex volumes. So visually what this looks like in this graph is, on the top row, we have the left hemisphere subfields. On the bottom, we have the right hemisphere subfields. And these 8 plots represent the 4 subfields within the hippocampus. And these 6 represent the 3 subfields outside of the hippocampus, but still within the medial temporal lobe. Again, these are the 5, subfield regions that were significantly correlated with maze accuracy. Moving on to looking at hippocampal subfield thicknesses and its relationship with maze accuracy. This is the table that we have with all of the correlation coefficients and their p-values. And again, taking into account sex, age, and total intracranial volume, we see again that left dentate gyrus thicknesses are significantly correlated with maze accuracy. Visually, what that looks like is, on this graph, we have the hippocampal subfield thickness for left dentate gyrus on the x-axis, and on the y, we have proportion correct, which is maze accuracy score, and we have a significant positive correlation. Nextly, we have the 3 other medial temporal lobe subfield thicknesses to look at. And when correlating that with maze accuracy, we see that the right perirhinal cortex is significantly related. And then also the left and right parahippocampal cortex and their thickness were related to maze accuracy. Visually, these are what the plots look like. So overall, what we see is that these subfield regions could be very informative in looking at their relationship with navigation accuracy in the maze, or just generally navigation ability. So moving on to looking at behavioral results. So this is just information about navigation ability and not looking at subfield volumes. We did some correlations between our navigation measures. So correlating the maze accuracy score with the questionnaire scores, we have the questionnaires on the left column, and then we have the n value, the correlation coefficient, and their respective p-values. Just to note, this is for the full data set of 107, but of course, some participants were excluded due to missing data. So to start off with one of our questionnaires, which is asking about video game experience for the participants. We had a couple questions listed on the questionnaire, but we pulled out 2 that were of interest for this research, which looks at the hours per week that an individual spends playing a video game that involves learning a virtual environment. So when looking at this score, which is again the hours per week score correlated with proportion correct in the maze, we see that there is a significant correlation between these variables. But interestingly, if we're looking at an individual who spends 0 hours a week learning an environment virtually through a video game, there's large variation as expected in navigation ability. But as an individual increases their hours per week score, on average, they score higher on the maze accuracy task. Nextly, when looking at video game experience with the units of years of experience, we see a similar result, where as one increases their video game experience in years, they also increase their maze accuracy, and this is on average. And lastly, we had video game experience as an entire questionnaire, so we created a composite score, and we see a similar result where there's a significant positive correlation between these variables. The next questionnaire on this list is the roadmap test, and this is a perspective taking task where participants were shown a route from a map, and then they were told to write down the turns of the route, so either the right or left turn, and this is done under a time constraint. So we found no correlation between the roadmap test scores with maze accuracy, and this is likely because the roadmap test is a perspective taking task, but within the maze, the participants were virtually looking at the maze in a first person point of view, so they probably didn't need perspective taking. Nextly, we have Santa Barbara Sense of Direction Scale. So this is a self-reported measure of just general spatial and navigation abilities. And in this in this questionnaire, the participants saw some statements such as "I get lost easily" or "I like to look at maps," and then the participants would rate how true that is for themselves. So looking at this plot, we have better perceived sense of direction as a lower score, and then a higher score would mean a worse perceived sense of direction. So when putting that on the x-axis and correlating that with proportion correct on the y, we see a negative correlation of 0.24, and this is a significant correlation that we were finding, which means that on average, if a participant has a lower SPSOD score, which would mean better perceived sense of direction, then they would score higher on the maze accuracy. Next, we have the spatial orientation task, which is another perspective taking task. And we also have handedness, which were both not correlated with maze accuracy. So moving on, we have some exploratory results for sex differences. And first, again, looking at just the navigation ability questionnaires in the maze, we did t-tests and between the males and females, and we saw a significant difference in the means between males and females for the maze accuracy scores, the video game experience scores, SPSOD scores, spatial orientation task, and handedness. So pretty much everything except the roadmap test, we saw significant difference in the means between the sexes. So specifically, the males tended to score higher on average than the females on all of these navigation measures. This is actually consistent with previous literature that has shown male advantage in navigation performance. And also we wanted to look at sex differences in our data, which we're looking at the subfield volumes and its correlation with maze accuracy. So looking at the same plot, but then plotting them by sex with the gray representing males and then this purple magenta representing females, we see that males tend to perform better on this maze accuracy score compared to females for all of these subfield volumes. We also looked at subfield thickness for hippocampus and also mediotemporal regions that all show pretty much similar results. So with that, I want to start by concluding with our main findings, which is that the subfield volumes within the hippocampus as opposed to total hippocampal volume might show some relationships with navigation in the healthy young adult population. Also, our exploratory analyses of sex differences show that males on average perform better than females in the navigation tasks. Some limitations to this work is that we did notice some premature cutoffs of the MTL regions within the high resolution T2 MRI scans, but we went around that by looking at not just volumes, but looking at thicknesses as well, which is another valid measure to look at critical size. And we also have a relatively small sample size to look at sex differences. However, this work is still in progress. We're still doing analyses, and we have 107 participants who completed the maze task, and we also have their T1 and T2 scans to look at their brain volumes, and we have to manually segment their hippocampus to be able to look at that, but we do have a full data set that has not yet been analyzed. Generally, we do have challenges with segmentation as well, since there's no single protocol to segment the mediotemporal lobe, and there's many that are out there that a lot of researchers use. It's not very consistent, but we did our best, and we worked with other researchers from other universities who have more experience in segmentations to help us with our manual edits for the segmentation. For future directions, we hope to recalculate our score for path efficiency, which would be another measure of navigation. Currently, we have a score that only includes correct trials, but we also want to include incorrect trials that would look at how efficient a person is at navigating from one location to their end destination. We also want to correlate other navigation measures, such as the ones from the questionnaires, to cortical volume and thickness. And then we also want to look at hemispheric differences as well as sex differences once we have our full data. So with that, I'd love to acknowledge some individuals. A huge thank you to Dr. Elizabeth Chrastil for all the help, support, and guidance throughout this entire project, as well as the other members of the Spatial Neuroscience Lab. I also want to thank Spatial Cognition again for this amazing conference, and then I want to thank our funding agencies. With that, this is my works cited. Thank you so much for listening, and let me know if you have any questions.
Thank you, Alina, and apologies to Dr. Chrastil for mispronouncing your name earlier. That was a fascinating talk, and as somebody who neuroimaging is very dear to my heart, I appreciate the challenge of how difficult it is to segment tissue in that region. It is a really, really tough task. So congratulations for doing that. And there are no definitive way to do it. Many groups struggle with this. But you mentioned the London taxi driver study, which of course is a famous one now. And it made me think of a study actually published in 2019 by Weisberg et al. And one of the co-authors is actually sitting there with us now with her hand raised. And I was going to invite her to make some comments about this study. So Dr. Newcomb, what say you?
Oh, well, Alina mentioned our work in the introduction, and of course it really looks at total hippocampal volume. So it's fully consistent. In fact, I want to congratulate you and Liz for leading the charge to look at hippocampal subfields and sort of nearby structures. I do have a question about not including retrosplenial complex. I realize that's kind of a little not there somewhere else, but it's been shown to be very important for navigation. I think there are some behavioral findings that suggest it may be linked to individual differences in functional studies. So I just wondered if you thought about that, and is it impossible, or what was your thinking there?
Yeah, definitely. That's a really great question. I think some projects within the lab are looking at retrosplenial cortex. I don't know too much about it, but I think that it's definitely really involved in navigation just generally, and it would be really interesting to look at. I just haven't looked at it for this project yet. But yeah, I think it would be interesting to look at for the future.
Yeah.
Okay, thank you. Thank you, Nora, for those insightful comments. So, Alina, I have a question too about an imaging question for you. And I'm thinking about what other changes there may be in the brain that obviously, you know, if there's got to be some sort of expertise that develops, and people have different levels of skills with navigation. And I'm wondering whether turning to the white matter might be give you something to grasp and hang on to. And I say that because a while back there was a study by Heidi Johansen Berg from Oxford, and she was looking at people learning to juggle. Now, juggling is also a spatial skill. It's not like spatial navigation, but they looked at also the white matter tracts, and so regions that are linked, for these sort of spatial abilities, and they found some gold in them, their hills there. So I'm just wondering whether, have you guys thought about doing white matter tractography or diffusion weighted imaging, and then going on to do tractography at all?
Yeah, that's a really great question. If I'm not mistaken, I think that is kind of like our next projects. So we do have some data on the DSI imaging, but I don't know if Dr. Chrastil can answer.
I can chime in. Yeah, we actually we've been doing a lot of work with the diffusion weighted imaging of DSI scans on everyone. So we actually finding some pretty cool stuff. So we're writing a paper right now. So check it out soon. But yeah, definitely a lot of the outputs of like fornix and limbic system stuff. So it should be cool.
that sounds really terrific. So everybody's got to watch for that when that comes out. So good luck with getting that into press. That's really, really great. And we've got some time for. I've got a speculative question. Since
Wait a minute,
nobody else...
Sarah Fabricant had a question in the chat.
Oh, I'm sorry. Oh, yes. She has a video gaming question. Go ahead, please, Sarah.
Don't worry, I'm the complete naive, non-neuroscientist, non-brain person. I mean, I have a brain, but I hope. So here's my naive question. I just wanted to say what a great presentation in the sense also being really inclusive for non-specialists. So this is fantastic. Your graphics and your explanations were really helping me to follow. So thank you very much. I have again, naive question, probably. It kind of sounded like, oh, yeah, you're a good video gamer, then you're good, you know, you're fine. So I was wondering if your task is more suggestive of, you know, first shooter games than actual navigation. And of course, I'm saying this with a really devil's advocate kind of background. So, and we talked a little bit the other day about, you know, the realism of the spatial setup of actually navigation and in a virtual reality setting. And I'm not, you know, questioning if you know, how greatly the walls are designed, or if, you know, finding a painting by, what was it, Van Gogh or something is sort of suggestive of navigation. Just in general, it sounds, you know, like in the sense of the task, it looked more like, and your population, I assume, are college kids, I guess, to say it sort of flippantly, and there's a proportion of males, and of course probably that it correlates with males just playing more video games than females. So maybe you can talk a little bit about this, how suggestive, you know, the video gaming is really here at work or not, or I'm just curious to know more about this.
Yeah, that's a great question. I'm also really curious about that as well. So one thing to note is that these were just correlations. So we did find a correlation between the video game questionnaire, like composite score, as well as the maze accuracy. So one reason for that could be that they're both virtual environments. So an individual in the maze is navigating through a virtual environment and not a physical, like, environment, like when we walk outside. So that could be why there is a strong correlation. Another thing that you mentioned was sex differences. So we definitely want to look into that further. So I took a class on gender in video games during undergrad, and a lot of what we studied was how males tend to play more video games, but they also have better spatial abilities. So it's kind of like, are they related? Is one or is one causing the other, or is it just a correlation? So this is all really interesting to look at and think about. I don't want to say, I don't want to predict anything wrong, but I think it is really interesting to look at, and we hope to dive deeper into sex differences when we're able to analyze our full data set.
Thank you.
Yeah, of course.
And Alina, there's no shame in making predictions that are wrong. I've been doing it for 40 years and I'm still doing science. So, you know, I encourage you to make predictions and make mistakes. It's part of the fun. We have bets in the lab, and we have prizes for when you know, people get things right and wrong. So I have one more quick one before we go to break, and then the poster session. And I was thinking about muscle memory when we navigate, because as we take these navigational tasks out of the lab, we're kind of missing one important dimension, you know, of walking around, wandering around, and kind of that experience of navigating the route itself physically. To what extent do you think that plays a role in maybe influencing the findings that you might be getting in the lab?
Yeah, well, for our lab, at least, we have different ways to measure navigation. So one is a desktop virtual maze environment where the participants are just lying in the scanner, they're not physically moving, but we also use VR. So the participants walk around in a space, like an open space, and they're looking through the VR, but they're also able to walk around. So that kind of gives us a better understanding of vestibular movements and how that applies or is important to navigation learning. But at least for this part, or at least for this study, we had a desktop environment, so we didn't include that information, but it would also be interesting to compare, you know, like the results from a desktop setting to a VR setting.
Yeah, that's exactly where I was going with that, because then that may explain, you know, some of these interesting gender differences, for instance.
Right.
I don't know, but it you know, it's a fascinating subject, and my other half and I like to argue about this all the time. So on that note, thank you so much for a very stimulating talk and beautifully presented as well. And so right now, all that's left for me is to thank all the speakers. Thank you for people for asking questions, and thank you all for being here. We take a break for 15 minutes, and at 17:15 we return to a poster session. So everybody get caffeinated, and we'll see you in the poster session. Bye-bye now.